Thank you so much to Jeremy and the rest of the Twist team for giving me the invitation to come and present today and share some of our work. So what I'll present today is really, in many ways, a journey through time, application of different genomic technologies to a really important clinical problem. Had the benefit, I guess, of seeing that evolution of different technologies over time. I've been working in this disease area for more than 20 years, in fact. And it's been an amazing, I guess, journey along the way and having the opportunity to look at these different technologies. Okay, so, whoops, we've gone the wrong way. Okay, so I guess a fundamental question that's asked when someone presents with a new cancer diagnosis, where did that cancer come from? This is a really important question, both for the clinician in terms of deciding on the best treatment options, but also for the patient as well. So this does occur in about 1 to 3% of all cancer diagnoses. It's probably reducing in time as our diagnostic methods are improving, but it's still a significant problem. In Australia, according to the Australian Institute of Health and Welfare, this represents just over 2,300 patients a year. So that's his 14th most common cancer diagnosis, but it's the 6th most common cause of cancer-related death. And as you can see in the figure shown here, this is the survival, 5-year survival, and it's really down where lung, pancreatic cancer and mesothelioma is. So patients progress quickly and naturally they would have metastatic disease. This represents very aggressive types of a solid malignancy. So without a lack of a definitive primary site, this really can restrict treatment options. It's really the clinicians are left with a best guess, I guess, of what to treat the patient with. But it also can have implications in terms of access to treatment as well, as most treatments are really only registered for particular disease types. As I mentioned, impacts patients significantly as well, creates great psychological distress for the patient and morbidity perhaps with extensive diagnostic workups for these patients. So what is CUP? Well, I guess through historical autopsy series, and this data was collected some time ago, so the fraction of cases within particular cancer types may have changed over time as our diagnostics have got better, but it generally represents aggressive solid cancers, carcinomas predominantly. And I think the lung cancers still represent the largest fraction of cases, but then made up of gastroenteropancreatic malignancies as well, genitourinary tract tumours, and in Australia, I think we have an enrichment of skin cancers as well, given the predominance or the frequency of skin cancers within Australia. The reasons for why they present like this can be multiple. Perhaps it's just a regressed primary that could be caused by clearance by the immune system. And we know that this occurs within skin cancers like melanoma and Merkel cell carcinomas, where they have a very high mutation load and they're very antigenic. The primaries can be small and clinically unidentified, and I guess we're getting better with this with the introduction of new imaging modalities like PET. But typically there's many cancers that become undifferentiated or transdifferentiated as they progress and acquire different mutations. The program of the cell changes. They lose many of the features that are recognised by pathologists to recognise what that cancer might be. Some of these tumours also represent rare cancers, or rare subtypes of more common diseases as well. So in 2014, we started a clinical genomic study, a national clinical genomic study called SUPER, Solving Unknown Primary Cancer. And this SUPER, I guess, version 1 went from 2014 to 2020, and we're employing these two different assays prospectively in patients to see how these assays may impact patient care and whether or not they can be effective in resolving diagnosis, but also directing treatments. This coincided with a dedicated CUP clinic being started at the Peter Mac by Linda Malishkin, a medical oncologist. But we are unique in that we've also got a PI, Penny Schofield, who is a behavioural scientist, and she's very much interested, I guess, in the psychosocial impact of CUP as well. And with myself and David Botel, we've really led the genomics side of this. So as a team, we developed SUPER, and it's been a very successful program of research over many years now, and has attracted funding from a number of different funding agencies. So the first test that we started applying was gene expression profiling, and back in the day this was microarrays. So this started for us back in 2001. We were generating our own microarrays back then, and my PhD in fact was on developing a gene expression diagnostic using microarray technology. And really the principle of this is that a cancer cell, even though it leaves the primary site of origin and metastasises to another part of the body, it doesn't entirely lose that cellular programming. And really this is what pathologists are relying upon for a diagnosis, the morphology of the cells, and plus with the use of immunochemistry, looking at specific proteins that are expressed on those particular cancers, most tumours can be resolved with histopathology in combination with the clinical picture. Gene expression profiling allows us to expand the number of targets we're looking for to a whole transcriptome, in fact, and we can use machine learning to develop classifiers that then classify an unknown cancer. And by looking at non-primary metastatic tumours, we can get a very high accuracy using this particular technology or approach. And then it's very difficult, I guess, in validating in cups because the true origin of the cancer is never known, but in some cases the primary will become evident in time. We call these latent primaries. And 78% of cases in this study that we published in 2015, we found that the tissue of origin predicted by gene expression was consistent with the pathology or the clinical picture. So this test was actually developed into a commercial assay by HealthScope. Unfortunately, HealthScope was taken over a short time later and the test was discontinued. I guess that's a common problem with development of diagnostics. Not only does the test work, but I guess the business model has to work as well. So this approach, we were one of the first to show this, or to describe this approach, I guess, in the application in CUP. But subsequently, a number of different tests were developed, and I'm showing a few here. The CUP guide was the one that we developed with HealthScope, but a number of others were developed using real-time PCR. Some were using microRNAs from Rosetta, and this one's a qPCR as well, or a microarray, sorry, this one. And EpiCup, I'll talk a bit about that later, that was using DNA methylation profiling as an alternative analyte. So the accuracy is pretty similar across all of these different tests, and many of them attempted to validate within these latent primary cups as well, showing a similar result. So we know that this test is likely to be better than immunochemistry, and there was papers to show that it was, but it hasn't been accepted into clinical use. There was 2 randomised control trials that tested this approach, and they found that by directing treatment based on the predicted tissue of origin compared to a randomisation to an empirical chemotherapy regime, there was no difference in survival outcome. There's a lot of criticism of these trials in that they didn't have access to some of the more newer treatments like immunotherapy and many targeted therapies for cancers like lung cancer. So this may have contributed to that, I guess, that null result for these clinical trials. So currently these types of assays are recognised in guidelines, but they're not strongly recommended. Of course, the second approach is to look at targeted DNA sequencing and to look for actionable mutations. And a number of studies have also looked at this in a larger series. And Foundation Medicine have conducted several studies and now involved in a large-scale clinical study or clinical trial called Capisco. In fact, they've stopped recruiting now, and the results from this trial, it's an umbrella trial that will be reported later this year at ESMO, I believe. And just looking at the numbers of patients that would, I guess, be allocated treatments within that umbrella trial in Capisco, we get around 30 to 40% of cases you'd find an actionable mutation. So, as opposed to the gene expression profiling, these tests are now recommended within NCCN guidelines and ESMO guidelines as well, although these tests are not rebatable within Australia for this particular indication. I guess what we've been championing for the last few years is that you can use DNA mutation profiling also as a diagnostic to help resolve the tissue of origin. So this study here we've just published late last year. This was led by a PhD student in my lab at Tara Posner. We went back and looked at the super series, and we went through each case with a fine-tooth comb with a pathologist to really see where each of these genomic tests might have been useful for resolving diagnosis. And just at the start, just through review, retrospective review of pathology. In fact, the pathologist felt that they could resolve the likely primary site in nearly a quarter of cases without the use of genomics. So I guess this sort of indicates that it also depends on the experience of the pathologist and knowledge that they have, that they might be better at, one pathologist might be better at resolving cups than others. All these patients were recruited across many sites in Australia, so the referral patterns, I guess, they were referred based on being cup and what the impression was of the clinician and pathologist at those sites. So we were left with 3/4 being what we called true cup, and then we looked at molecular features. We looked at all immunochemistry and molecular features by panel sequencing. We looked at mutational signatures that we could detect in panel sequencing, and panel sequencing can be useful for this, as particularly within a hypermutated tumours, those associated with smoking and UV exposure, corresponding to lung and skin cancer respectively, pathognomonic driver mutations, and even detection of oncoviral DNA. In summary, we found that 31% of true cups, DNA mutation profiling was helpful in resolving diagnosis, and 15% of true cups, RNA expression was helpful. So this looks quite actually a lot different to what had been previously reported, and I think it's because we're looking at a much harder group here in terms of resolving it. It suggests that gene expression profiling isn't so great for those really hard cases. So now we're turning to expanding, I guess, or making the test more comprehensive. And I'm generally referring here to whole genome sequencing, but that could also correspond to more transcriptome-wide or methylome-wide analyses as well. So we have to look at a cost-benefit analysis here of what it costs to do whole genome sequencing compared to panel. It is more expensive. You do get more diagnostic information though from a whole genome. The time, I think they're almost equivalent now, to be honest. If you're set up to do whole genome sequencing, you have the analytics, you have high performance computing, you can turn these around quickly. It's actually the curation time that takes longer. There's just a lot more to look at. The technical reliability, certainly panel sequencing is still superior to whole genome, only in that the types of material that we get access to, in Australia at least, is generally FFP core biopsies. It's very challenging samples to work with. So now we've expanded to 3 different studies. They're not all genomics related, and we've been very successful in attracting some MRFF funding. So I won't go into these other 2 studies today. One's looking at a new PET tracer called FAPI, and the other is an implementation study looking at the optimal care pathways for CUP that we developed. So I'm just going to focus here on SuperNext. This is comprehensive genomics today. There's really 2 aims to this. It's to compare the diagnostic yield and the clinical significance of whole genome sequencing over panel sequencing, and the second is to explore circulating tumour DNA. And we're collecting, this is overlapping with, I guess, the first version of Super. We did attract some more sites, and we're encompassing more states now, but we're getting samples from as far afield as Darwin. We've got 2 sites in Tasmania. We've got many regional sites also in Victoria. It's very important to allow access to this type of technology and diagnostics to patients in regional areas. And we're actually generating 4 different molecular reports now, and plus a histopathology report. I haven't put the 4th report up, which is circulating tumour DNA. We've just started using that in anger. But each patient will get a panel, whole genome, and this couple report that I'll talk about in a moment. So just to give you an idea of the overview, in addition to this testing and reporting, we're collecting clinical data, baseline, 6, 12 and 18 months follow-up, patient-reported outcome measures, and we're having clinical questionnaires to really measure the impact, how this testing changed the clinician's view. And we're also looking at, we've got pathology questionnaires as well, pre and post genomics, to see how the genomics has helped the pathologist. We're doing a health economics analysis, and we've got a biobank that we've developed. We've even tried generating some cup organoids. To do all this, we need a really large team of people, and this is really spread across 2 different centres, but we're all co-located in the same building, which is quite convenient. This is the VCCC building in Parkville. And we have clinical coordinators, we have people, project managers, and RAs who are important for the logistics of actually just getting hold of these samples and communicating with each of the sites. We've got two major genomics platforms, one at the Peter Mac and the Peter Mac Pathology that's doing the panel sequencing, led by Andrew Fellows and Stephen Fox, but I guess a commercial arm to the Peter Mac Pathology called Genomic. We've got a very dedicated team of curators, both within panel and whole genome. These guys just work tirelessly, poring over data from panel and whole genome and curating that and getting it ready for reports, which are all discussed at a weekly molecular tumour board. So just some data, I guess. This is preliminary data along the way. And so not surprisingly, whole genome sequencing has a greater diagnostic yield. And here I'm just splitting up to ACMG tiers. Notably that tier 1, we don't capture any more. We haven't caught any more in terms of actionable targets, therapeutically actionable targets within whole genome of a panel. There's a lot of information here which really relates more to the diagnosis of these cancers and perhaps resolving what is truly a germline variant versus somatic, because we're using a somatic-only panel or a tumour-only panel. One of the things we have noted was when you're using these tumour-only panels, you do see some discordancy in this tumour mutation burden, and really this reflects populations who are not well represented within databases like Nomad. In fact, we get a number of cases coming from places like Darwin where we presume that perhaps some of these patients have Indigenous ancestry and we're getting calls of TMV above what they should be, and essentially it's because there's germline variants not being filtered out of that data to call that TMV. Real-world experience using FFP, where we have to use FFP for most of our cases. And I guess we're being brave and using this for whole genome. Many other sites, such as in the Netherlands, at Hartwig, or in the UK, they won't use FFP. Probably they're wise not to if you don't have to. But it's going to take a long time to change practice to have routine collection of fresh tissue material for diagnostics. So you get the good, the bad and the ugly. But we do have mechanisms to filter out those cases or QC the DNA. We've got good at doing this, so we don't try to sequence things that just aren't going to work. It can be variable across sites as well. Peter Mac's very good. I've blanked out the names of the other sites. I don't like to name and shame those other sites that might have bad FFP. I don't think it's their fault. It's just that these processes weren't designed with molecular testing in mind at the end. In summary, 64% of the patients get whole genome, 90% get panel. So we get a much higher yield in terms of the numbers of patients we can sequence with panel. Okay, so just returning to this Cupper report, I'll try to move through this quickly in the interest of time. So with the looking at genomic landscapes of different cancers, we now have a very good understanding of the molecular features of these cancers in terms of driver mutations, copy number profiles, structural event profiles, etc. We look at SNV96 mutational patterns, and I mentioned the smoking and the UV, for instance. And then we also have this regional mutation density. This is not something that's widely known, but you won't get even distribution of mutations across the genome. It's very much dependent on the epigenetic landscape of the cell of origin and the DNA repair processes that are operable in their cells. And you can use this information as to profile tumours, and this has great tissue of origin, information that you can use in a machine learning sense. And so we've had a great collaboration with this group at Hartwig Medical Foundation. Hartwig actually based in the Netherlands principally, but the lead of their bioinformatics is in Sydney, Peter Priestley, and he's got a fantastic team who've developed this tool called Cup prediction algorithm. He's taking a lot of these features that I just spoke about, these SNV96 trinucleotide signatures, the genomic position, or the regional mutation profiles, and then DNA features, and these could be passenger and driver mutations. He's also recently incorporated RNA classifiers, looking at gene-wise, just total gene expression, but also alternative splicing. They can all be combined, and we're training on a very large data set from Hartwig. They have more than 6,000 whole genomes that they've sequenced there, and plus he's pulled in the ICGC data as well. And so to develop this a very accurate and very useful tool, this all runs on a cloud-based system on Google Cloud. This is just sort of a raw output. all the different cancer types, and then you get a likelihood score. And you can see in this case, the high likelihood score across all the different classifiers, individual classifiers, and then a pool, so you get a DNA or an RNA combined, DNA combined, and then DNA and RNA combined as well. And everything lines up like this. You're pretty sure that that's likely to be the diagnosis. And so just on validation, this was in the Hartwig set, getting very high prediction accuracy, 91.3%. But then you have a likelihood score above 0.8 and it goes up further. And we just did a small series on our own cases and we found similar results. So we report in anything above the 0.8 threshold, and anything below that we say is unclassified. So just giving you a case example, this is an undifferentiated malignancy, an 83-year-old female patient presented with an isolated lymph node disease and under her armpit. And cancer imaging, there's no evidence of other disease. The pathology is really this sort of sarcomatoid features. They couldn't resolve it any further. The immunohistochemistry was really non-informative, so it wasn't expressing any of these common melanoma markers, for instance. So there's a differential diagnosis which included melanoma, but it was undetermined. And so we ran Cupper on this case, and it did come back with a high confidence prediction of melanoma. Had a very high TMB, very strong UV signature, and mutations which are commonly found, specifically NF1. We don't know what this BRCA2 mutation could have just been a passenger mutation in this context. A B2M deletion might mean an escape mechanism, and perhaps potentially the patient might not respond to immunotherapy. That rarely deters the clinicians from treating with an immune checkpoint inhibitor, which is really standard of care for this disease. One thing to note that the prediction from DNA was very high, but RNA was low. I guess it comes back to that point that RNA isn't very good in some of these undifferentiated tumours. So just a summary, I guess so far I mentioned previously with panel sequencing, we think we can resolve around about a third of these true cup cases, whole genome sequencing, and combined with all the decision making with pathologists and clinicians, we think we can get that now to around about 2/3 of the cases resolved. So let's just go to methylation, because really that's the reason why I'm here talking to you today. And just to give you some background, I guess, I was involved with a study which was led by Manel Estella in Barcelona. He developed a tool or a test called EpiCup based on Illumina methylation arrays that performed very well, as I previously mentioned. But very few people have actually compared across these different modalities or different test types. There was a case from the master study in Germany that recently published, and they compared gene expression versus methylation. They didn't look really at mutational profiling. Very low correlation between these two different things, or two different tests. So I'm very keen to understand this, because we've got whole genome sequencing with Cupper, we've got the gene expression, and we've done a very good job at digging deep in each of these cases. We have a very good understanding of each of these diseases. It would be good to layer on methylation now. I guess we've been prevented from doing this up to date because we get very small tissue biopsies. Often there's not even enough to run whole genome and panel, never mind looking at methylation. But if with this new, I guess, EM-seq method, which has been marketed with Twist really for capture, this is really opening up possibilities of using low input material. And this can be used for FFP, but also for cell-free DNA that I'll mention. Okay, so the other thing we're interested in is cell-free DNA as well. And we know that 10 to 40% of CUP patients won't have enough, there won't be insufficient material to do your standard tests. We know that tissue retrieval itself can take up to 11 days, or it's actually an average of 11 days. Taking a simple blood draw would shortcut that. And perhaps we could even use a blood test much earlier in the diagnostic workup as well. So in fact, we've employed Cupper on some of our cases where we've got high tumour fraction in the blood, and it can work. And there's a good example here of a patient where Cupper is classified as esophageal gastric cancer with a high likelihood match. And the profile looks very similar between tissue and blood. But it doesn't work for all cases, and there's many reasons for this that I won't go into today. But I think really what Cuppa, the most recent version of Cuppa, is doing well. It's doing well because you've got DNA and RNA. We need another orthogonal analyte there to include, and perhaps DNA methylation could be it. So we get very high yields of ctDNA from cup samples. So the average is around 7.6 nanograms per mil of plasma, and almost a third of cases you'll get really high tumour fraction. But running Cuppa as it is not going to work on 70% of those cases. 15% of tumour fraction is really the threshold that we use for tissue. So we've done some pilot work on using twist methylome capture, and we've looked at cell lines. We've looked at a few fresh samples. We actually have in the pipeline a lot more data, but I couldn't get that ready for you today. And we've done ctDNA analysis as well. And this is really the performance metrics. I guess the one thing I want to highlight is that the ctDNA samples worked pretty well. In fact, they probably work better in our hands, I guess, for this pilot than it is for cell lines and fresh. That may be a reflection on our processes, and we're still getting accustomed to using this assay. So I suspect that we'll be hitting more of these twist recommended metrics as we become more accustomed to using this test or this assay. This is just looking at correlation of EM-seq versus EPIC array for these cell lines. Very high correlation between those two tests. And really what we ultimately want to get to is to be able to classify a cancer based on its methylation profile. So we just thought, let's just throw all the data together with TCGA data. TCGA is a collection of different cancer types. There's 30 different cancer types in TCGA collection. I haven't used all those in this analysis. But it's EPIC array, well, it's Illumina methylation data. It's not twist data, so we've got a cross-platform I guess, problem there. But all the cases from fresh tissue actually classified correctly based on what we thought they are. The ctDNA samples did not classify correctly. We certainly need to do more work in trying to normalise the data. So this was just the ctDNA samples that we ran. We did choose the best cases, I guess, for this, in that they are high tumour fraction cases, between 16 and 42% tumour fraction in blood. This is estimated by doing low-pass whole genome and using this tool called iCore-CNA. This is just showing you the types of cancers that we tried to profile. And we selectively chose some neuroendocrine carcinomas. The lab has a strong interest in neuroendocrine carcinomas as well. So we want to include those as we have intentions of using this method also in that disease context. But what we were able to do is just differential methylation analysis between the neuroendocrine and the non-neuroendocrine tumours. And indeed, we could find many DMRs that are specific to neuroendocrine. So I'm just highlighting some of these neuroendocrine types of transcription factors. This is sort of just a higher resolution shot, I guess, looking at the context of those CPG sites, whether they're within the promoters, UTRs, exons, etc. And so this is encouraging that we can do differential methylation within cell-free DNA. And I'm sure we're going to get this tissue of origin classification to work. In fact, I'd be very surprised we didn't. There's many others that are doing something similar to this. For instance, Grail, with their gallery test, is relying upon DNA methylation for tissue of origin as well as early cancer detection. So just in summary, I've shown you that Cuppa is really a devastating disease that causes significant morbidity and mortality. It's really a composite of solid cancer types, predominantly carcinomas. In Australia, we do get a good number of skin cancers that are not carcinomas. So I've shown you SuperNext, or the SUPER program. It's really a national clinical genomic study, probably one of the longest running clinical genomic studies in Australia. And I've shown you that whole genome and transcriptome can be useful for tissue of origin classification. I think there is good reason to use whole genome in this disease. It may not be true for all cancers, and perhaps we'll have more discussion about that later in the panel discussion. Cupps have an atypical RNA transcriptional profile. I think this really necessitates orthogonal different tests to be used for classification. We know that DNA methylation is a viable option for multi-omic testing, and this methylome capture kit can be applied to cfDNA, which is really quite great. So just to finish with acknowledgements, there is many people to acknowledge. There's teams, as I mentioned, at the University of Melbourne and the Peter Mac, and this goes from the super team itself to the genomics platforms, the curation scientists. I really want to thank my team for preparing some of these slides, which were really coming to me only yesterday. So I thank them for their efforts. Of course, to thank the patients who contributed samples to the study. We've got a dedicated consumer group for this super program, and to all the collaborators at different sites. And also, of course, to our sponsors, including Twist, who have really supported this study. And I'll finish there. Thank you.